So, good morning, dear colleagues. We are starting our final day of the school, and the last lecture will be given by Professor Mariam Ramazanova, Organization of Radiopharmaceutical Production. Very important topic. So, the floor is yours.
Hello, everyone. I'm happy to be with you today. And today I will read the lecture about the organization of radiopharmaceutical production. And let me say a few words about myself. Okay. My name is Mariam Ramazanova, and I have a big experience since 2009 in manufacture in radiopharmaceutical production sites. It was in-house production and manufacture sites, licensed manufacture sites in Moscow region, and we manufacture fluorine radiopharmaceuticals, fluorine 18 and carbon 11 methionine radiopharmaceutical for positron emission tomography. In my experience, I have and design qualification, and now I'm working as an expert in design and organization of radiopharmaceutical sites and facilities. Today we will talk about the GMP rules for radiopharmaceutical production and radiopharma sites because it's most important to radiopharmacy part, and when we say about the organization of radiopharma production or radiopharmaceutical production, it's need to understand that all rules, it was GMP rules. Our lecture plan for today, you can see on the slide, we say about the radiopharmaceuticals, terms and definitions, regulatory documents, pharmaceutical quality system, risk management, about the personnel, premises and equipment production, about radiopharmaceutical processes, and the most important process, aseptic production and processes, quality control, documentation, and about the self-inspections, audits, computerized system, CAPA plans. All of it, it's part of the organization of your radiopharma production. First of all, we start of the terms and definitions. We should understand that we all say in this and understand the terms, right? First of all, radiopharmaceuticals. It's a group of pharmaceuticals, drug, medicinal formulation containing radioisotopes. First of all, radiopharmaceuticals, it is a medicinal products which, when ready for use, and contains one or more radionuclides. We have two another terms, preparation and manufacturing. Preparation sometimes used for the in-house production, but the manufacturing production, it's a production, quality control, and release and delivery of radiopharmaceuticals from active substance from starting material. And it can be manufacturing in-house production in the medical centers or some university centers, or the full cycle manufacture production stay like a plant. Then we go to the next slides. It's need to understand that radiochemicals, it isn't radiopharmaceuticals. Radiochemicals, it's starting material for the radiopharmaceuticals. it can be raw material, but radiopharmaceuticals, how I say, it's a medicinal product, and this product, all processes for this product should be validated, and radiopharmaceuticals should be prepared with GMP rules. GMP, it's the most important part for radiopharmaceutical and for marketing authorization for them. We should have licensed promises. We should certified personnel, and all our pharmaceutical quality system should be with GMP rules. But both radiochemicals and radiopharmaceuticals can be manufactured in the same center. And how I say, radiochemicals is the starting material for the radiopharmaceuticals. When we say about the organization of radiopharma production, we should understand what we want to achieve as a result. Because now we have a lot of different radiopharmaceuticals. It can be radiopharmaceutical for SPECT or for PET, or now most popular radiopharmaceuticals for therapy. And maybe you know about the new type of radiopharmaceuticals, theranostic radiopharmaceuticals, when we use the molecule with different radionuclides for the therapy and for the diagnostics. Then when we understand what we want to achieve as a result, it's radiopharmaceutical for SPECT or for PET or for therapy, we need to understand which molecule we want to use for our radiopharmaceuticals, because when we plan our production site, it's different regulations for the different radiopharmaceuticals. For PET, it's one type of rules. For SPECT, it can be another type of rules. But the therapy, it's long live radionuclides and need more protective or more promises. And when we organize this production site, it's need to know what we start to do, because we can't prepare fluorine 18 in the same room with iodine 131. It's a different type of radionuclides, and we need two different another promises for them. Okay. Next slide, and about types of radiopharmaceuticals forms. Why it need to understand when we prepare our organization plan of radiopharmaceuticals? Because it's important for the promises and for the equipment. 90%, I think 90 or 80% of radiopharmaceuticals, it's this solution for intravenous administrations. It's a sterile products, and we need special promises for the sterile production promises. It's a grade A classes room, clean room. But for solutions for internal use, it's another type of radiopharmaceutical form. No so strong rules, and we have a class D or class D area productions premises or sites. Another types of radiopharmaceuticals forms, it's capsules or microspheres. Capsules and microspheres can be prepared in class D area. But if microsphere, then we use in the solution for intravenous administration as a starting material. We need class B or class A area for the preparation for this type of radiopharmaceutical. In that case, need to understand in the start of your project what type of radiopharma forms you will be prepared in your site. Four categories of radiopharmaceuticals. Radiopharmaceutical preparation, when we prepare from the starting material and use synthesizers. When we use a radionuclide generator or radiopharmaceutical precursors, and then kit for radiopharmaceutical preparation. These four categories for radiopharmaceuticals need to know when we plan our production site. It's a starting information of your design. Now, very interesting part of organization you can see on the QBD approach. Quality by design is a strategic process for development and manufacturing. QBD approach is described in ACH documents, and they can show for us all life cycle for the pharmaceutical. Our radiopharmaceutical is a pharmaceutical part, and we can use this approach from the starting, from the research and development, from the R&D part, and use it's still the finished our production for radiopharmaceuticals. What's about the quality by design? Its approach starting from R&D, and then when we use this approach, we can understand all our process from start to finish, and all critical parameters, all critical materials, all important point we can find from the start, from the beginning, from the research. And then when we started our production, we can understand processes, and our quality will be higher, and product knowledge and product specification all time will be in the specification quality. In that case, QBD cannot be without GMP. GMP is a part of QBD approach. It's a step of manufacture. And GMP, it's a quality management system for pharmaceutical products. It's a part of our quality by design approach. When we say about the GMP part, it's it's a production and in the control, quality control for the radiopharmaceutical. It could be clinical trial authorization. It's a part of the marketing authorization before we start to use our radiopharmaceuticals to the patients. And we can start to use for the patient, our radiopharmaceuticals only after marketing authorization. For all of this reason, we need some plans. Now we can use some instrument from Microsoft MS Project when we start our plan, some roadmaps from the start of our project, from the design, then starting building and production, marketing authorization. But we need to understand every step. One of this plan you can find general radiopharmaceutical implementations schema. This plan from guide EIE, good practice for introducing radiopharmaceutical for clinical use. We can use this plan because here you see the first step, identifying radiopharmaceutical, then conduct literature search. We need to understand what knowledge in the world for this radiopharmaceutical we have. Then formulation question, identify synthesis, prepare submission packaging, plan, and very important, confirm plan with the regulators, obtain financial resources. Financial resources, unfortunately, it's a very important part for all of us because we need to understand that our pharmaceutical, when after we develop it and it will be used in medical center and patients and financial parts. It's a very important part for each project. Sometimes project not started because financial so big and we can't start it. But now nuclear medicine and radiopharma production site, it's a future time. What type of radiopharmaceutical manufacturing we know in the world? It's type of radiopharmaceutical manufacturing from ENMM. We can have a large-scale industrial manufacturing. It's a big plants where we can produce radiopharmaceutical, and small-scale in-house hospital radiopharmacy units. The type of this in-house production, it can be hospital radiopharmacies. It can be nuclear medicine departments, research PET centers, and in general, any healthcare establishments. For positron emission tomography, very popular in-house hospital production, hospital radiopharmacies, or nuclear medicine departments, because the fluorine-18, the most popular radionuclide for the positron emission tomography, have a short shelf life, only 2 hours, and we prepare this radiopharmaceutical in in-house hospital radiopharmacy. What type of methods of the production? For the small-scale production, we use radionuclide light generators and cyclotron production. If I return here. If we have a small-scale in-house hospital radiopharmacy unit in our nuclear medicine departments or hospital radiopharmacies, we can equip our centers with cyclotron or with a department with radiopharmacy with radionuclide generators. For the plants, it's a reactor production. Or sometimes cyclotron production for the large-scale production, where we can find information about the guideline of the radiopharmacy production for the small scale. For the big scale, it's only GMP rules. But for the small scale and radiopharma production, we can find in documentation from the European Association of Nuclear Medicine. They have a guideline on the current good radiopharmacy practice for small-scale preparation of radiopharmaceuticals. This document give steps for each type of small-scale preparation, site 1, site 2, and site 3. And about the European Association of Nuclear Medicine, it's a professional non-profit medical association and facility communication, which was founded in 1985, and they help to the organization with some guidelines in nuclear medicine, and they have a lot of information about the radiopharmaceutical, how we can use it, and for preparation too. Another document, guideline on quality risk management for radiopharmaceuticals. It's a very useful document in practice because when we start our organization sites, in each step we should prepare the risk assessment, and one of the instrument of risk management, it's this guide describe the instruments for the quality risk management. Another type of guides which can help to organizing of radiopharma production sites. It's International Atomic Energy Agency guidelines. They have a lot of types. I think you know them. Here on the slide, I show the quality control of in the production of radiopharmaceuticals. Now we can use it because here we have information about the type of equipment, about the methods, and if you can't find some information in pharmacopoeias, you can see it in these guidelines. And about the big document, GMP, in European Union, GMP rules, it's Etralex 4.0. Here on slide, you can see all chapters and annexes from the Etralex, from the GMP, European GMP rules. All these chapters can be implemented for the radiopharmaceuticals. And when we can start our production or organizing our production site, first of all, we can read this document or the GMP in your country. Because in each country we have the same rules, the same GMP. The World Health Organization, it's a starting document, and all of these documents are harmonized with a document in each country. FDA documents for current good manufacturing practice, it's CFIA 21, and in Eurasian Economic Union we have GMP rules, decision number 77. All these rules is a requirements for the radiopharma production, and they have chapters about describe each part. Why is GMP, GXP, and I show you the QBD part, how the life cycle of the radiopharmaceutical, because we can see in the schema from the start to the finish which type of documentation we can use in each step of our organization part from development. Development now it's ICH documents. Sorry. Then when they start the preclinical investigational, it's a GLP rules. When the clinical trials, it's a GCP rules, good GCP rules. Then marketing authorization, and when started our production GMP rules, GCP, good storage practice, GDP, good distribution practice, and then patients. In each step, we have some requirements and rules, and all of it's a quality assurance system for each organization. We can see GXP compliance. It's means, and you can change for the part of your compliance, and it will be a good distribution practice or good manufacturing practice. You can see on in literature CGMP or CGCP. C it's mean current. It's a current rules. I said about the financial resources, and this slide describe what type of financial resources we need for the radiopharma production. It's most expensive, maybe. It's a clean room facility because we have a different type of clean rooms, class A, B, C. And D. It's a special ventilation system, different ventilation system. We can't use only one ventilation system. It should be different. We should prepare some gowning for the personnel. It's maybe not for each personnel, but it's important part. Quality control laboratory space, hot cells synthesis model, quality control equipment. We have staff resources and the source for radionuclides, cyclotron, target, generator, or commercial supplier. Each type of this part, it's very important to calculate before start your design, before start your project. And another part of the preclinical and clinical trials, it's very expensive and sometimes a long period. And when we design our organization plan, we should remember about this stage too. Some features of radiopharmaceutical production. How I say, first of all, it's pharmaceuticals. And we say today about the GMP rules because it's pharmaceuticals. Small scale preparation. Our batches of radiopharmaceuticals could be 10 vials or 2 vials. Sometimes if we have about we have planned and maybe you know radiopharmaceutical Xofigo. It's a big batches for the radiopharmaceutical, more than 100 vials. But when we say about small scale preparation, it's really small scale because in big pharma we have 1,000 or more vials, and 100 vials. It's small scale preparation. For PET radiopharmacy, batch preparation maybe 5 or 10 vials for the batch. Another features, it's a short shelf life. Short shelf life. What it's mean? We don't have a big storage space in our plant or in our hospital radiopharmacies. Short shelf life means we should have a good distribution practice, and we should understand how this radiopharmaceutical will be distributed to the hospitals. It was be cars, special cars, or the airplanes, or some different types. Radiopharma production, it's aseptic process production, and because generally radiopharmaceuticals administered via intravenous route, and sometimes we have a final sterilization with autoclave, but this type of sterilization used in plants. In the hospital radiopharmacies, we use aseptic processes with sterilization filtration. Conditionally, radiopharmaceuticals released before all QC results because sterility. It's this test prepared 14 days. But flu desoxyglucose, if you know, the storage time for this radiopharmaceutical 12 hours or 8 hours, and when we get results of sterility, we don't have the radiopharmaceuticals. In that case, the special rules for the radiopharmaceuticals, and they can be released before all QC results. But it don't mean that we don't need to do it. We should do it, but we get this result after we administrated it to the patients. Sometimes radiopharmaceuticals produced just in time. It's in the hospital radiopharmacy departments, used for diagnostics, no side effects because small mass amounts, and they use fluorine or carbon-11 radionuclides for diagnostics. For therapeutics, potential side effects because we use a lot of metals radionuclide in these radiopharmaceuticals. And very important, it's radiation hazard for personnel. This is some features of radiopharmaceutical production. We should know them when we organize our radiopharma production site. Okay, these features we can see in each part of our pharmaceutical quality management system. Pharmaceutical quality management system include each block on the schema, and it's a part of the GMP rules of Yetralex or Russian European Union, Eurasian decision 77 GMP. About pharmaceutical quality system, the holder of manufacturing authorization must manufacture medicinal products so as to ensure that they are fit for their intended use, comply with the requirements of the marketing authorization or clinical trial authorization as appropriate, and don't place patients at risk due to inadequate safety, quality, or efficacy. It's important because we prepared our radiopharmaceutical for the patients. The final product which we get, it's the product for the patients. And the safety and efficacy it's important part for each of us because each can be a patient in the medical center. And all GMP rules and GXP rules, the goal of these rules, it's safety, quality, and efficacy for the patients. For this reason, pharmaceutical quality system include each part of life cycle of radiopharmaceutical, its preparation, research development, distribution, and continuous improvement. Pharmaceutical quality system element you can find in each step. When we start our production after pharmaceutical development, after technology transfer, we validated our processes and then check periodically with your plan how work your radiopharma production site. For this reason, we have CAPA management and change management and quality risk management instruments. When we assess and evaluate our pharmaceutical quality system and our processes. About quality risk management, it's instruments for each pharmaceutical production site and in radiopharma production too. Here you can see a paragraph from the ICH guideline Q9 about the quality risk management. It's important to understand that our product quality should be maintained throughout the product life cycle, such that the attributes that are important to the quality of the drug medicinal product remain consistent with this used in the clinical studies. When we started our manufacturing processes, we can evaluate our processes with risk management instruments. Which type of risk management instruments we can use? This type of fishbone Ishikawa diagrams. This diagram can be used from the research and development when we analyze our processes, and then in next stage of production site, when we have some problems with our processes, we can this instrument can help to identify a root case and the first problem, if we have some problem with process such as small yield, or we didn't understand why we can't understand sterile product. We can use the Ishikawa diagrams. Because sterility or bacterial endotoxin, it could be problem from personnel, it could be problem from the equipment, it could be problem from the starting material. Ishikawa diagram can help to find this problem. Another instruments, it's a failure mode effects analysis, FMEA method, and failure mode effects and criticality analysis, FMACA method. These methods with ranking, with the bells, we can use the number of priority of the criticality or risk and use this instrument for the next assessment. Okay, next slide. You can see on the schema each block, one of block documentation, risk analysis, another block it's a personal. For aseptic processes in radiopharmaceuticals common, very important which personnel will work in your site, because production of radiopharmaceuticals depends on people. In my experience, I have 3 different sites, and I understand that how you training your personnel and what personnel will work in these sites. It's very important, because we have small batches, we have a small shelf life, and if somebody forget add some chemicals or screw some tubing, we can lose all batches. Here, it's very important what type of personnel work in your radiopharma site. Sufficient number and qualification of people, yes, and clear roles and responsibilities. Maybe you know, qualified personnel, qualified person, then we say about this person. It's stuff which can give the responsibilities that when we use our radiopharmaceutical for the patients. Training, we should repeated training and validation for critical tasks, aseptic handling. For aseptic handling in GMP rules, we have Annex 1, where described all aseptic handling and media field test when we check how our personnel work in aseptic processes. All trainings should be documented and recorded. We should have training matrix. And we have documented procedures for handling with radioactive material and radiation safety production. What type of organization structure could be on a radiopharma production site? It's different departments. We can have a radiation research and development department, but if we have in-house production hospital only for the patients, this department sometimes have or not. Production department, it's very important. Quality assurance department very important. Quality control department, here the quality control lab or microbial lab. IT and technical department. Now it's very important because we have a robotic or automatized equipment. All module synthesis are automatic, and another part of GMP requirements, it's a validation of a computerized system which we use in production sites. In that case, IT and technical department, it's very important to have in the radiopharma production site. We have warehouses, radiation safety department, and qualified person or persons or responsible person if we have in-house production. Premises and equipment production. Premises and equipment protect product against environment. How I say, we use grade A and grade B equipment areas for the intravenous production, aseptic processes, and grade C, grade D premises for the laboratories. Radio protection, protect environment against product. For working in the radiopharma production sites, we use hot cells and shielded hoods. We should have qualification, installation, operational and process qualification for premises and equipment. And calibration for equipment, all measuring devices we should have with calibration. For production area, we have two approaches. First approach, it's a handling with open sources of ionizing radiation. We have two types, 1, 2, and 3, when we work with open sources with the radionuclides or radiochemicals. And the second approach is the clean room areas, classes A, B, C, and D for the radiopharma production with GMP rules. Here on slide, you can see the layout of in-house production radiopharmaceutical in hospital. You can see 3 zones. First zone, green zone, it's a reception area. Blue, it's a medical area. And yellow part, it's a production site with a cyclotron. You can see here the cyclotron, the laboratory, hot cells, the QC laboratory, and the all manufacturing steps should take place in the self-contained facility dedicated to radiopharmaceuticals. When you plan your radiopharma production sites on plans, you can see the flows, the roads of each type of materials, radiopharmaceuticals, personal, if it's a hospital patients, QC probe, and waste. It's very important that describe these roads, because we didn't have any cross-contamination between personnel and patients, between waste and raw materials, between different radiopharmaceuticals. And when we plan our production site, we should use risk assessment instruments to describe why we use this type of roads in our scheme. About the clean rooms, I'm return here. When we say about the hot cells here stay in the clean room area. The manufacture of sterile products should be carried out in appropriate clean rooms. Entry for personnel should be throughout the personal airlocks, where the personnel change clothes and going into the special type of clothes for the pharma production. Here you can see the ventilation system of the QC lab, and you can see pluses. Pluses, it's mean pressure inside the room. When you design and in radiopharma production or another pharma production, it's very important to understand the pressure inside the clean room area. And we use here risk analysis instruments to understand what type of pressure could be used for this room. Clean rooms used special materials. How you see on the photo, it's a special equipment inside with the stainless. This equipment should keep the disinfection and special special material for the work. About the clean rooms, we have a lot of documentation because clean rooms used not only in pharma, clean rooms used in the microelectronic production sites. And in this slide, you can see International Organization for Standardization, ISO, standards for clean rooms. About the classification, grade A, grade B, and the grade C and D, or class A, B, C, and D. These grades described in Annex 1, manufacture of sterile medicinal products, very important for aseptic processes. Grade E zone, it's a critical zone with the high risk operations. Here the filling zone, here the open primary packaging, or making aseptic connections under the protection of first air. For these classes, we have special hot cells for the production area, and the production of radiopharmaceuticals for in grade A and grade C area you can find. For sterile solution for intravenous injections, what type of equipment we use? Dispensing cell, it's a class A area, pre-chamber, grade B area, a hot cell grade C, where we can put synthesis model, and airlock where the personnel change clothes and go in. It's grade D, and then grade C area. In table 1, it's from GMP rules, where we can find the requirements for each classes. For particle concentration in each area, for classification each of type of clean rooms. How it control? We can control and monitoring our clean rooms with different measurement instruments. What we should control? We should control pressure inside the clean rooms and clean room area, humidity, temperature, and particle counter we use for the control of concentration of particles inside each area. We can use particle counter inside the hot cells, or we can use control environment for all clean rooms and each equipment monitoring system, which combine all equipment for control temperature, humidity, air velocity, particles, or sometimes microbial control. Another part of Annex, it's manufacture of radiopharmaceuticals, Annex 3. You can find in GMP rules in European Union, in GMP rules in Eurasian Union, and the CFIA 21, Annex 3. Accordance with Annex 3, manufacture of radiopharmaceuticals, sterile radiopharmaceuticals may be divided, which are manufactured aseptically or terminally sterilized. How I say, a lot of radiopharmaceuticals prepared in aseptic processes. Terminally sterilized, it's could be radiopharmaceuticals with iodine or with lutetium or with radium, but all type of radiopharmaceutical with fluorine 18 or carbon 11, it's aseptic processes with sterilization filtration. For manufacture of radiopharmaceutical, risk assessment may be applied to determine the appropriate pressure difference, airflow, direction, and air quality. Risk assessment, it's a very useful instrument in your organization site. In case of use of closed and automated systems, we use a chemical system and robotic system for the preparing of radiopharmaceutical. And prior to start of manufacturing, assembly of sterilized equipment and consumables, tubing, sterilized filters, or sterile closed and sealed vials to sealed fluid path must be performed under aseptic conditions. It means that all our starting materials should be sterile if we prepared aseptically our radiopharmaceuticals. Hygiene and governing personnel. On the slide, you can see the personnel, the staff of radiopharma production site. Different parts and for governing for different premises, and hygienic and hand disinfection standard for each organization. Sanitation and hygiene are vital in every aspect of the manufacturing processes. It covers anything that can cause contamination, from such as personnel, the premises, equipment, containers, or production material. All potential sources of contamination should be identified and eliminated with a comprehensive sanitation and hygiene program, because this part is very important in radiopharma production. Every production site should have cleaning, sanitizing, and decontamination procedures. What type of disinfectant we can use? We can use peroxide or some different disinfectant which are registered in each country. For aseptic hot cells for grade A and grade B area, we should use sterile disinfectant. For grade D, we should control the concentration of our prepared disinfectant for the cleaning, sanitizing, or decontamination. For the control of microbial inside your production facility, we can use different instruments. It's microbial air samplers. The requirements of the microbial monitoring includes air, surface of clean rooms and zones, equipment, clothes, personal gloves, primary packaging materials, microbial contamination of compressed gases, purified water, and disinfecting solutions. Why it's important? How I say, we can get the results of sterility test after the patients get the radiopharmaceuticals. In that case, microbial control, it's a very important part of the radiopharma production sites. Contamination with microbial contamination can give us a good quality for bacterial endotoxin and sterility. The sources of the microbial contamination, it's could be air, personal, water. The first of all, personal and starting material. Next type of premises and another department, very important department, warehouses or warehouses area for the in-house production. We should have 3 warehouses or warehouses area. It's for raw material, for our final product for radiopharmaceuticals, and another type, very important part, it's for labeling and printed material, or it could be some scarf or some area inside the warehouses. In the schema, you can see the way, the road of the raw materials and why it's important. It's a big GMP part, raw materials. We get materials from the vendor. First of all, we should audit our vendor. It's a vendor selection part. For all starting materials and critical components, only qualified vendors should be used. Vendor qualification can be established by an audit, by responses to quality assurance questionnaire, or simply based on experience with the supplier. In any case, vendor qualification should be documented. It's a very important part. Audits of a vendor, it's a GMP part. Then we received our starting material. It's a reception of raw material. Then we sampling from this batch of raw materials, and before we get the result of our samples, these materials stay in the quarantine. All type of these materials identified, marked with labels, is in quarantine. And after we get results of QC our samples, we can move our raw materials from the quarantines to the main warehouse of raw materials. If we get not good results for our raw materials, and it's a no in specification, we should reject this material, and in our warehouses we should have this field for rejected materials. The same schema for the final product for the radiopharmaceuticals, but we don't have this vendor. Vendor, it's our site. But we have zone quarantine and warehouse of radiopharmaceuticals. This type of schema can be used for the radiopharmaceuticals with a long shelf life, as iodine 131 or iodine 123 radiopharmaceuticals. Okay, next. Production. It's a paragraph from the GMP rules. Production operation must follow clearly defined procedures. Each step in your production site should be described. Personnel should understand every step from the entry inside the building and from finished his work. The principle of production, each step of production should be described in your procedure. In this flow chart, in this schema, it's a technology process of fluodesoxyglucose. This process starts from the beginning, from the personnel, from the premises. It's a preparation of gowning, preparation of equipment, preparation of premises, and then we go to the cyclotron. Laboratory synthesis, and technology gases, which we use in our production schema. Here you can see every step from the cyclotron to the final product in the container for the hospital. Production equipment for the radiopharmaceuticals, how we say, we start from the cyclotron, and next step is the hot cells. Hot cells, it's a shielded workstation for manufacture and handling with radioactive materials. Inside the hot cells, we can achieve a class C or class B or class A grade. And inside the hot cells, we can put some dispensing system or module synthesis. PET radiochemistry systems module synthesis. Here are some of examples. It's automated processes and all type of module synthesis now uses cassette technology with GMP rules. You can see here the research and development model synthesis when we combine some radiopharmaceuticals and start to prepare some different syntheses. FastLab, it's FastLab or TRACIS models. These models are used for fluorine and metal labeling system for positron emission tomography, and now the new type of models, it's for therapy production. It's no laboratory production, it's manufacturing production when we produce radiopharmaceuticals with GMP rules. How we can put this radiochemistry system inside the hot cells? It's a two-module synthesis. Inside, when you plan your production site, you can understand how much hot cells you need for your your radiopharmaceuticals. You can combine and put two radiochemistry system or one. For target materials, we can use another hot cells, and for dispensing system, we can use different hot cells with a robotics system, which are designed with GMP rules. This system used single-use system materials. And for aseptic processes, we have sterilization filtration, and inside the kit of for dispensing, we can use the sterilization filters position 6 for on this schema. This dispensing system very useful because we haven't any handle operation. All operation we with computerized system. Next step of production radiopharmaceuticals, it's labeling. Labeling, you know, we can't label our radiopharmaceutical after we dispense. Yeah, and now a lot of manufacturer of equipment have inside the dispenser special labeling system for the labeling without visual contact, without handle contact. Requirements for the labeling you can find in the pharmacopoeias or the rules rules in your country. We should labeling the primary our vials, and after that we put our vials or our robotic system which we use put our vials in the special containers. It's the secondary container labels. Requirements for labeling secondary containers you can find in GMP guidelines, or you can find in your pharmacopoeia. These steps of radiopharmacy production could described in production schema, and critical process parameters should be accepted. Some examples of critical process parameters, it's temperature, activity, volume on the synthesis steps, pressure inside the hot cells. It's a critical process parameter because our production is the pharmaceutical production, and laminar flow, velocity, labeling. It's critical process parameters. Okay, next, aseptic processes. For terminally not sterilized productions, we should have we should product our products in class A clean area with background class B area. For terminally sterilized products, autoclaved or filtered radioactive solutions, class C area is recommended. We use terminal sterilization for the iodine 131, 123 radiopharmaceuticals. For fluorine 18 and carbon 11 radiopharmaceuticals, we use aseptic processes with sterilizing filtration. Sterilizing filtration used 1.22 millimeters membrane filters with special membrane. When you develop your process, you need to do some tests to validate your sterilization filtration and to understand what type of membrane we can use, because for the water solutions we can use polyethylsulfone PES membranes, but this membrane cannot acceptable for the solutions of PSMA, because PSMA fluorine PSMA content ethanol and ethanol and PES and don't good combination. For the PSMA, we can use another type of membrane filters. It's PVDF filters, types of the Kesivex from ABX solutions we can use in our practice. But for each type of filters, when we use this type of sterilizing filtration, we should do integrity test. Integrity test it's a non-destructive test used to predict the functional performance of the membrane filters. And for this tests, we can use special equipment. This equipment can include inside the hot cells with the grade A area. It's a bubble point test equipment. And for each batch, we should prepare this test before and after dispensing. About quality control. How I say, some radiopharmaceuticals may be distributed and used before all chemical and microbiology tests have been completed. And what type of release of these radiopharmaceuticals? First type, it's allowing transportation of the radiopharmaceutical under quarantine status to the clinical department, and then qualified person should conditionally certify the product before it's used and should finally certify the product after all the test results are obtained. A release of radiopharmaceuticals for the two steps before all QC results we get. Quality control department structure, it's another part of structure. Here could be the quality control laboratory, analytical laboratory, microbial or radiometric laboratory, promises for stability study, promises for reference and retention samples. When you plan and organize your radiopharmaceutical production site, we need to understand how much departments and which premises we need for quality control. If we have in-house production with fluorine, it's enough to have a quality control lab and promises for reference retention samples, or maybe an analytical lab. But if we have a big plant, we need all type of these rooms inside your manufacture. Which type of samples for quality control we get? What we control? We control final product. First of all, radiopharmaceuticals, our batch production. We can control intermediate products and in critical stages. Sometimes you can see active pharmaceutical substance or excipients, and we control packaging and printed materials. Where we can find requirements for the QC methods, it's our pharmacopoeias. We should choose testing methods accordance with pharmacopoeias. In each country, we have national pharmacopoeia. If in pharmacopoeia we didn't found some methods, we can find them in another pharmacopoeia or develop QC methods. If we develop our methods, we should validate this method, and only after that we can use these methods. If we have a pharmacopoeia method, we should verify this method with a pharmacopoeia. Okay, QC, quality control of radionuclides and radiopharmaceuticals. Which tests we do? These tests from pharmacopoeia. Radionuclidic identity determination, it's a method for half-life determination, gamma spectrometry, radionuclidic purity, dose calibrator measurements, and radiochemical identity. We for radiopharmaceutical, for intravenous injection, it's osmolality, sterility, endotoxin content. For all of radiopharmaceutical radioactive concentration, visual inspection, pH, specific activity, we use chromatographic methods and radiochromatographic methods. In this slide, you can see some of equipment for in-house radiopharma production site. It's isotope calibrators, different type of chromatographs, osmometers, pH meters. In different countries, we have standards for the equipment or for the in-house production. In Eurasian Union, we have standards for the medical centers and QC equipment for this center described in the national requirements. What type of work quality control should do? Another type, it's a stability study. Stability study, it's a study for all pharmaceuticals, not only radiopharmaceuticals, the aspirin and our fludesoxyglucose, the same here. But for radiopharmaceuticals, it's this study is not so expensive how for the big pharma, because we can understand our stability of our radiopharmaceuticals for the day or some days, because shelf life of radionuclide 2 hours for fluorine 18 or next day we can get the results of the stability of these radionuclides. Why it's important? Is the example with fludesoxyglucose. As you know, the activity the activity of fluid desoxyglucose for the patient to 250 megabecquerels. We can prepare this fluid desoxyglucose with a small cyclotron, which produce 100 gigabecquerels, or with big cyclotron, which prepares 700 gigabecquerels. And if starting activity from cyclotron is so big, we can see the autoradiolysis effects. And in that case, fluid desoxyglucose, which we get on the cyclotron with big start activity, cannot stable in a period of 12 hours because the autoradiolysis effect. The another example of the molecules of PSMA. PSMA, it's denaturated when we started from the activity more than 200 gigabecquerels from the cyclotron. In that case, it's this stage. It's very important for all of type of radiopharmaceuticals. Quality control of raw materials. What type of raw materials we use? It's not only chemicals. It's different parts of cassettes or kits for dispensing. It's gases, and in our pharmaceutical quality system, we should have procedures which described our storage of raw materials and sampling. Reference and retention samples. It's a QC part 2. For radiopharmaceuticals, we should keep our retention samples at least 6 months after expire. Samples for starting material, we should keep 2 years after we use this product, after release this product. In that case, we have enough space in your manufacture site or in-house site for keeping this material. Documentation, it's a big part. The GMP sometimes this abbreviation, good manufacture practice, but sometimes they say great mountain of paper. Because GMP, it's a lot of documentation, how I say, from the starting to finished. What type of GMP documents we should have in our production site? We started from the quality manual and the different SOP and master production document, master formula for each radiopharmaceutical which you produce on your site. Specification for each finished product, for raw and packaging material, and if you have AP, active pharmaceutical substance, or in bulk production. We should have documentation, testing methods, working protocol, procedures for identify labels. And for each steps, we should have records. And these records should be in paper, or can be automated in a computerized system. In that case, all systems should be validated. Self-inspections. When we working in our radiopharma production site, sometimes we should do self-inspections. Self-inspection provided by personal staff which work in your site. And we check all our GMP parts inside the production area. One of part, it's a qualification and validation. Qualification and validation are applicable for the facilities, equipment, utilities, and processes used for the manufacture of medicinal products. It's a GMP requirement that manufacturers control the critical aspects of their particular operation through qualification and validation. It's a big part of each GMP production site because qualification and validation qualified should be promises, all equipment which you use, gases, and if you have a grade A or grade B area, you should qualify this zone every 6 months. For the equipment, for each equipment should prepare design qualification, FAT, SAT, installation qualification, operational qualification, and process qualification. All your processes should be validated. If you prepare fluodeoxyglucose intravenous, module synthesis, synthesis of fluodeoxyglucose should be validated. Aseptic processes should be validated. For that case, we have validation master plan. Laboratory information management system can help to organize your production site. Here, the example of information LIMS laboratory information system Petra, but now we have another products, and we can combine all reports from the cyclotron, module synthesis, quality control equipment, and we can get electronic dossier for the batch and product release and batch records we get in time. But it's most important that all computerized system should be validated, and we have Annex 15 in Hydraulics in GMP rules about the computerized system and requirements for them. Next type of management, change management. When we organize our radiopharma production facility, in-house production, we should understand that we will change in next time something. Maybe our process will be changed, or maybe we can change our vials, or maybe we can change our promises in next time. We can't change it without I want to change. And I change. For this reason, for pharma production, we have change management. Every change should be documented, evaluated with risk assessment, and after that, we can decide about implementation of this changing. About the corrective action and preventive action, CAPA system. It's another part of control when we organize our radiopharma production site. The pharmaceutical company should have a system for implementing corrective actions and preventive actions resulting from the investigation of compliance, product rejections, non-conformances, recalls, deviations, audits, regulatory inspections, some findings, and trends from process performance and product quality monitoring. It's important part because all processes, production sites sometimes have some deviations. And if we have some of deviations, we should document it. If some yield not good, we have a specification specification diapason from 50 to 90%, and we get from our radio chemical synthesis 75. This 75% will be deviation, and we should start the process of corrective and preventive action. We should open kappa plan and started investigation why we get this small yield. Here, the type of application of corrective action and preventive action system throughout the product life cycle. This type of GMP part, the kappa plan, and kappa management, you should prepare every year and assess at the end of the year what type of deviation or findings was identified, and you should have a plan how to correct or prevent this. This final slide. Thank you.
So thank you very much for the very detailed presentation. I'm quite sure that this information will be helpful and useful for our participants. Questions? Yep.
Thank you for the very nice presentation. My question is regarding the radiochemical identity, I should say. I may have misunderstood, and I'm referring to the radionuclidic identity, because I know for some cases where we irradiate something in the cyclotron and we get a product, we let it decompose, and we find that some longer half-life nuclide was present in such a concentration that while the product that we anticipated was there, we couldn't measure it because of the much higher counts. So is there any way to assess these kinds of impurities, or do we just accept it as the limit of our measurement technologies?
Oh, okay. I understand your question. Maybe you say about the starting material for the radiopharmaceuticals or for AP, active substance from the reactor, or like this. When we prepare our specification for the QC, for the control of radiopharmaceutical, we should include for the final product all requirements with the pharmacopoeia. For starting material, we can justify our specification limits or test, which we will do. For example, for lutetium chloride from the reactor, we have really big activity from the AP. From the starting material, and we put the vial with the AP inside the module synthesis without information about activity for the milliliter or something like this for radiochemical identity. But for the next steps of technology, purification or next steps in synthesis, we should get the final radiopharmaceuticals, and we can check it in the final radiopharmaceutical. Your question may be part of the research and development part. When we use small activity and we have a model of modeling of our synthesis in the small activities and try to get a clear product with good specification parameters, with good radiochemical purity.
Questions? There is one question online. Just a moment. So, why to place the synthesis model in hot cells? Is it for maintenance of the synthesis model, or it has an impact on the final product?
About maintenance for the synthesis module.
Yeah, in hot cell.
And hot cell. For the hot cells, we have a technical area without clean room grade. It's the backside of the hot cells. For the module synthesis, we should plan our production, work in our production site that we have days for the maintenance, week or maybe months, when we maintained our equipment. Or another part, if we have a room, technical room, where we can move our module synthesis and maintain, but it's I don't see such examples.
And the activity in the synthesis model is very high, so that's why it's in hot cell.
For this hot cells and for these modules, we have prepared a plan when we stopped our production site, waiting sometimes, but we didn't get cold module synthesis. It was still hot, but we can maintain it, such as a cyclotron. Cyclotron all time is hot, but we maintained it every quarterly.
If no more questions, so thank you again for your great presentation.
So, dear colleagues, please take your seats. We prepared some questionnaire. So, Dr. Gilman.
Andrei. So guys, let's take just a short look on our tests, on the results of the tests. So we will start with introductory tests. So the first question was about what are isotopes, and most of you answered correctly. So it's just this pair of uranium atoms with different masses. So we know that isotopes, they're nuclei of the same element, but with different masses. All others, they are not isotopes. They're either different forms of the same element, or they're in neutral or charged form, or it's isobars but not isotope. Somebody wants to join. Okay. So the next one is about matching the type of particles with their description or definitions. Yeah, alpha particle contains 2 protons and 2 neutrons, not 4 neutrons. So beta minus is just electron, negatively charged, and gamma radiation, yeah, simply electromagnetic radiation. So again, 2/3 of you answered correctly. That's I would say it's in advance that this school, the participants of this school, got the highest marks among the other schools. So it's already the 7th or 8th school, and you had the highest mark. And that's why it was really difficult to reach the higher level, but it will be in the last slide. So the third question about the definition of not definition, but equation of how the activity is changing. And almost all of you answered correctly. It's changing with time with the exponential law. It's decreasing with time. The fifth question about the how activity is changing with time after 2 half-lives. The correct answer is B. It's the activity will decrease by 4 times. And the most difficult question was about identification of the products of the decay of different parental radionuclides, but still more than half of you answered correctly. That's very nice. Which kind of ionizing radiation has the largest penetration range? Here, some of you probably was confused with the penetration ability and ionization ability. So the highest penetration ability has gamma, and some answers was with alpha, but yeah, I think it's just confusion of ionization ability and penetration ability. The 8th question is very easy, because all the answers are correct, because in nuclear medicine we assume not only radiopharmaceuticals, but any kind of application of ionizing radiation in nuclear medicine. So for diagnostics, it's only X-rays or gamma rays, but the answers with beta may be used as correct, because if we are talking about beta plus, then it annihilate and produced gamma radiation. So I took these answers as correct, but in principle they are not correct, because again, type of radiation that detected is gamma in this case. But okay, I count it as correct as well. So here it's usually when we're talking about the radiopharmaceutical, it can be these parts of the radiopharmaceutical. I didn't count here the correct answers because, of course, most of you mentioned the radionuclide, but different parts can be or cannot be in the radiopharmaceutical. So I didn't count this. The Coulomb barrier. We have a Coulomb barrier when we have interaction of two charged particles. Yeah. So if we have neutron with no charge or photons that have also no charge, so we don't have a Coulomb barrier. But protons, they're charged particles. That's why then they interact with the nuclei. Then of course there is a Coulomb barrier. But again, quite a lot of people answered correctly. The last question in introductory test was about the detection of gamma radiation. What types of diagnostics use this gamma radiation detection? And oh, yeah, I missed this one. Okay, and it is positron emission tomography, single photon emission compute tomography, and planar scintigraphy. But MRI, it's magnetic resonance investigation, so there is no gamma radiation. Some of you mentioned this, so that was not correct. And now the final test. Here, I changed just the order in the first question, so it was again confusion. I saw that some of you just picked the first answer as it was in introductory test, but so we yeah, pay more attention on the terms. Here now it's after 3 half-lives. Yeah, it may be a bit confusing because it's 2 in the magnitude of 3, so it's 2 multiplied by 2 and multiplied by 2, so it's decreasing by 8 times. Here now in the final test, more people answered correctly, so that is pretty nice, because in introductory test it was around 60%, now it's 75%. And the next question was specifically about radiopharmaceuticals. So now it's not in nuclear medicine in general, but in radiopharmaceuticals. And here, for therapy, we can use alpha and Auger emitters. I would say practically we don't have proton emitters for application as a radiopharmaceutical. And the fifth question was also about diagnostics, but with radiopharmaceuticals, not in nuclear medicine in general, but specifically with radiopharmaceuticals, and it only positrons and gamma radiation. So again, it's a bit confusion because the only changes in question, I would say one word. So previously it was nuclear medicine, now it's radiopharmaceutical. So again, pay more attention. The next question, the sixth, was also confusing for, I would say, many of you, half of you. Most of the people, not most, but half of the people answered 50 millisievert because they divided just by 2. We increased distance by 2, and thus people just divided 100 by 2. But actually, we decreased the dose rate as a squared distance if it is single source or point source of radiation. So it is 25 microsievert will be. Again, repetition of Coulomb barrier, and many of you answered correctly. So the new topics: what are the highest cross section of nuclear reactions? And thermal neutrons have the highest cross section. So quite a lot of people. I was surprised because it's not the easiest question. What types of diagnostics based again on gamma radiation? More people answered correctly. That is good. About the protection of intellectual property. Yeah, don't say anybody about your unique inventions if you don't want that it will be used improperly. So yeah, the correct answer here is no. So the cross-section of neutron induced reactions, again, it's quite a difficult questions. And it was, I would say, the competition between two answers, either this or this, and the correct answer is B. So it's 1 divided by just the velocity of the neutron. So the radionuclide generator here was honestly a bit surprised because quite a lot of answers was about apparatus, but we know that radionuclide generator is just a system where we have equilibrium between parental and daughter radionuclide, and the difference between B and D that here it was mentioned that parental radionuclide has a shorter half-life. That is incorrect. So in radionuclide generator, parental radionuclide has a longer half-life. So who makes the final decision? Most of the answers were correct. It is only the government of the country, IAEA or OECD and EE can recommend something, but cannot make the final decision for the country. And radionuclide purity, quite a lot of people remember that it is fraction of total radioactivity in the form of the desired radionuclide in radiopharmaceutical. And that I was also pleased to see that most of the participants answered correctly about the three main principles of radiation protection. Yeah, it's again justification, a LARA principle, and dose limits. So by comparing the beginning and the end, we see that at the start it was a really high percentage of correct answers. And even if we had quite, I would say, more difficult questions at the end, this still 80%. But yeah. I would say that it is really a great achievement because the questions were much more difficult than in introductory tests. So yeah, you are all very nice students. So that's it from my point. Now, Andrei.
Please.
Может, куда-нибудь туда?
Подскажите, а где он хуже стал? Такой вот мы встаем здесь с Ириной, соответственно, к нам уходит человек, мы разворачиваемся, фотографируемся с дипломом, и он уходит. А вам, наверное, удобнее будет вот оттуда где-то? Просто свет не будет? А, ну вроде он сдвинулся.
Мне кажется.
Да, да, конечно. So, dear colleagues, I want to introduce our partner, my friend Irina Sarkisyan, who co-organized with me in this school. So, Irina.
Yeah, guys, hello. Happy to see you all here. What I want to say, I'm happy to be here not only as an alumni of Moscow State University, but as a part of Rosatom's team. And first of all, I want to say thanks to our colleagues from Moscow State University and for sure to Olga, especially because I know how hard to organize any event, especially for colleagues, partners from all over the world. So the last but not the least, for me, it's very important to lifelong learning. And I think everyone should get information from everywhere and through all the life of their lives. So for us, it will be the best indicator if you will use that knowledge that you get from this course on your daily basis, on your work, on your studies. So anyway, I wish you good luck and hope to see you again. Thank you.
Yeah, thank you very much. Irina, from my side, I want to say thank you all of you. So you are right now participate in our Moscow State University family. So and welcome once again in our schools next year. So very glad to see all of you next year in next schools. Thank you very much. So and let's start. Ceremony. So I want to ask to come to me, Yusuf, Oluwatosin. Yeah, it's you. Ayoda. Yeah. Rashid, come to me, my friend. Congratulations.
Thank you very much.
Thank you too. So please.
Thank you.
Thank you. Shalpan Nazarkulova. Yeah. So you. Congratulations. О. Niota Tegoni era Subbiza. Yeah. Is it correct? No. Yeah. Congratulations. I need Olga because she knows all of your first and second name. Yeah. Okay. Matthias Fodor. Congratulations. Galata Leonage Nalaka. He's not here. Yeah. Yeah. Congratulations. How are you? How do you feel? Carlos Sebastian. Sebastian. Congratulations.
That's from myself.
Yeah. Steven Mayuka. No. Steve. Congratulations. Thank you. Mikhail Birulia. Congratulations. Юлия Столбунова. Congratulations. Николас Атуми. Congratulations. Дмитрий Казимирский. Глория Гифт Мугиза. Congratulations. Ayşenur. Asma Yali.
Thank you.
Доктор Гермак. Congratulations. Srivinesh Punaram. Yeah. Congratulations. It's fine. Okay. Николай Чебукин. Лизавета Устинович.
Спасибо. Спасибо. Спасибо.
Максудур Рахман Удой. Congratulations. Ксения Менюк. Поздравляю. Илья Домбровский. Я поздравляю. Валерий Минич. Поздравляю. And Самуэль Яв Азимах. Congratulations. So congratulations, all of you. Now we plan to make a photo all together, maybe here. So, and after that, I want to ask you to put some sign on the poster of the first floor. So please, Yulia, can you help us with pens and something? Yeah, okay.
Сейчас. Сейчас здесь?
Да, я думаю, здесь, да.